We are on the subject of molecules that make extracellular matrix. So far, the molecules that we have discussed are basically either carbohydrate molecules or protein molecules to which large carbohydrate molecules are attached. Now let's shift our attention to another very important component of extracellular matrix, the protein component. And we are going to start here by discussing collagens. They are the most abundant proteins in animals, in multicellular animals. They make about 25% of the dry weight of the total protein content. So let's look at collagens. Collagens are basically a family of fibrous proteins. So there is not a single collagen molecule. There are many different types of collagen molecules. We collectively refer to them as collagens, but as you will see, these different molecules of collagen have very different properties and they play very different roles in the physiology of our cells and our bodies. They are found in all multicellular organisms, animals I should say. You don't have collagens in plants, which are secreted by the cells of connective tissue and also some other cells. Collagen molecule is generally long and stiff, so it does not have elasticity. Elasticity in the tissue is basically a function of a different type of molecule, which is called elastin. We'll talk about that later in this course, but for now, we're talking about collagen, and collagens do not have too much elasticity. These molecules are triple-stranded helical structures. Remember, DNA is a double helix. Collagen molecules are triple helix. In this triple helix, the collagen peptides are called alpha chains, and they wind around each other in a rope-like superhelical fashion. So here you can see on the diagram, here is a single molecule of collagen. This is a single molecule of collagen, and it is helical. 3 of these helices come together and form the structure, a molecule of collagen. The green balls basically represent one collagen molecule, the red another collagen molecule, and blue a different collagen molecule. So 3 of them are coming together to form this triple helix. Collagens are extremely rich in proline and glycine. Both are important in forming the triple helix. Proline is important because of its ring structure, and it stabilizes this molecule in a helical conformation. And glycine, as you know, is the smallest amino acid. Here the small balls, red balls, are basically glycines. And when you go to the triple helix, you will notice that the smaller red balls, the smaller blue balls right here, and also the smaller green balls, they're pointing towards the inside of this larger structure, the triple helix. So generally, every third residue in this molecule is a glycine molecule, and this basically permits this molecule to form the triple helix, the glycine residue or the glycine amino acid, which is part of this polypeptide and therefore is referred to as residue. The single chain is about 100 amino acids long. Each amino acid here is represented as a ball. Here you will also notice another thing, that one red ball is basically X, the other red ball is Y. And we have a small ball right here. The small ball is always a glycine, pretty much always a glycine, as we have shown it here. The X basically is a proline, and Y is, in majority of the cases, hydroxyproline. The 3 alpha chains, they wrap around each other again, and glycines, the smallest amino acid, is in the center of this triple helix. So if we had a different amino acid, say if it was a larger amino acid, anything other than glycine, you can well imagine from this photograph that it would disrupt the triple helix, because these molecules, these other red balls will be pushed out, these blue balls will be pushed out, and this triple helix will not be stable if the small ball was anything other than a glycine. There are 25 distinct collagen alpha chains that have been identified, each encoded by a separate gene. Theoretically, this means if you have 25 different collagen proteins that cells can make different, and if you can combine them in different combinations, you can have thousands of different types of collagen. We don't have thousands of different types of collagens. We only have 20 different types of collagens in our body. The reason is not all alpha chains, it is not that any alpha chain can combine with any alpha chain. So the total number of collagens basically in our bodies is 20 different types. The main type of collagen found in connective tissue is type 1, type 2, type 3, type 5, and also type 11. Type 1 is the principal collagen, is the main collagen that makes collagen in the skin and also bone. Type 1, 2, 3, 5, and 11 are the fibril collagens or fibril forming collagens, which form these rope-like structures that we have seen in our previous slide. Other collagens can also have a helix, but they do not form fibrils. For example, collagen 4 we will see in basal lamina, for example, rather than forming ropes, it forms a mat, a sheet, a thin sheet like a sheet of paper. So it's not forming ropes. So different collagen molecules, different structures, and they help different tissues physically. So here, for example, this is the precursor alpha chain. It will form the triple helix. The triple helix, each of this triple helix will go through a modification. We'll talk about why this modification is important. Basically, the ends of this triple helix are removed, and the collagen molecule basically assembles into collagen fibril, and collagen fibrils, several collagen fibrils assemble together to form this collagen fiber. Here I would like to point out this, basically you can see this, there is a striation in this molecule. This is due to regularly placed collagen molecules in the collagen fiber. So they're about 67 nanometer apart. So this distance is 67 nanometers, and this staggered placement of collagen molecules basically gives the striated appearance to collagen fiber or collagen fibril. If we were to look at a collagen fibril, we'll see a photograph, electron micrograph of that, and it will also be striated. After the collagen is secreted in extracellular space, the collagen molecules assemble into higher order collagen fibrils, which we saw in the previous slide. And these are thin structures, roughly 10 to 300 nanometer in diameter, and many hundreds of micrometers long in mature tissue, and they're clearly visible in electron micrographs. I'll show you an electron micrograph in the next slide. But let me mention this, that collagen fibrils often aggregate into larger cable-like bundles, which are several micrometer in diameter, which can even be seen with the help of a light microscope. So let me show you that electron micrograph. Here, for example, is a cell which is producing collagen molecules. Please also notice, I don't know how clear it is in this photograph on your screen, but you can look at it up on the web, that this cell has abundant rough endoplasmic reticulum. Remember, all the cells that are making a lot of the cells that are secreting a lot of molecules, they have abundant endoplasmic reticulum. Here you can see these collagen fibers and this collagen fibrils, and they're organized into bundles. And also the point here in this micrograph is they're at right angles to each other. This is the orientation of this particular microfibril, collagen fibril, and these collagen fibrils are coming. Just imagine they're coming out at you out of the screen, so they're at right angle with each other. So different arrangements in different tissues. We will have a lot more to talk about how collagen is arranged in different tissue and who arranges collagen into a specific orientation.